Hi, welcome to Chem 1093. My name is Ryan Webb, and today we're going to be going over my video presentation covering the hierarchical structures of macromolecules. Macromolecules are large molecules necessary for life that are built from smaller molecules. There are two basic ways to build a macromolecule. First, it's going to be by the monomer. Monomers are labeled as building blocks. And we're just going to draw a little bit of blocks together that are not connected. Then we're going to have a polymer. Polymer is the units of the building blocks coming together to form the macromolecule. And a chain. The basic ways that macromolecules are derived from are from carbs, lipids, proteins, and nucleic acids. First, we're going to talk about carbs. Carbs are composed of mainly carbons, hydrogens, and oxygens. They're labeled as monosaccharides as individuals, or they become polysaccharides in a chain link sequence. Mainly, they're used for short-term energy use, and they come from things like breads, pasta, and sugary beverages. Next, we're going to be covering lipids, also known as fats, and they're going to provide us with two basic structures of having a glycerol and a fatty acid. Like I said before, fats contain two major components. It's going to have glycerol, which is the head, and then it's going to have the fatty acid, which is the tail. The glycerol and the fatty acid are also going to be, this is going to be polar, and this is going to be non-polar, making the glycerol a hydrophilic component, which is going to be accepting of more polar ions and molecules, and the fatty acid, which is going to be hydrophobic, which rejects most of the water, oxygen that's trying to flow through the membrane. Alright, so furthermore, amino acids are going to be the way that they become structured and combining with one another. The essential basic building blocks of our amino acids are just themselves, the monomers, amino acids. So they're just built into a chain, like that, and then becomes a secondary process. And these are bound together by hydrogen bonds through the amino acids kind of coming and grouping better together. And there's going to be two types. There's going to be the pleated sheets, which are kind of just overlay crooked ends. And then there's going to be the alpha helixes, and that's just one strand, kind of like a loose spaghetti noodle, that will eventually combine with the other side to become a double helix once we get into nucleic acids. The third part is going to be tertiary. Now tertiary is going to occur and present itself from attractions between the pleated sheets and the alpha helixes, so I have two different colors, but they're not as tightly bound together as eventually when we get to the quaternary. But they're just going to be kind of in the area of one another, such as that. Kind of just hovering. Now the final stage of what a protein is the building blocks of monomers. They're going to come to the quaternary portion, and that's going to be a protein consisting of more than one amino acid chain, and they're going to be tightly bound together in a better sequence of those hydrogen bonds that you're originally talking about. And such things as like an egg yolk becomes high protein. You can see it happen, and its structure unravel once you cook an egg protein, but once it's bound in there in the nucleus, you can see everything as a yolk is just one protein itself, and that's a resemblance of what that is in a very smaller subunit. Finally, we're going to move on to the fourth step. Unfortunately, I had to erase the amino acids portion, but we're going to move on, and I'll do it a different color in case you want to go back and take a clip art of the previous one in blue. But nucleic acids is the fourth component, and they're going to be main components of what we call our DNA and RNA. And those are very fundamental structures of the fueling process towards our amino acids being able to open up those enzymes and create substrates for which our molecules can break down our macromolecules. So nucleic acids, their monomer is going to be called a nucleotide also. And their components are going to be derived from creating coding and traits of genetics from the RNA and the DNA strand. And there's 3 parts to this. So there's 3 parts. One is going to be the sugar molecule. 2 is going to be the phosphate group. And 3 is going to be one of 4 nitrogen bases. Now, the nitrogen bases for DNA are going to include T, A, C, or G, whereas on the RNA sequence, it's going to be a U, uracil, and then A is going to still connect to T, and then it's going to be C and G as well. Alright, so a little bit more covered on nucleic acids are going to be those components I wrote down here for the amino acid start and stop codons. The T, the A, the C, and G for DNA, or the U, the T, the C, and G for RNA. And those components match up perfectly with one another to create stop and start codons, which begins and ends the transcription process of when DNA translates RNA within our genetic sequences of our body. Just in case you kind of didn't catch what I said before earlier, the DNA sequence is going to have the T, which is going to result in the RNA sequence having an A, the A for the U, the C for the G, and then vice versa going through. But remember, DNA will always have that TA, and then once the A is going to be translated, it's going to have the RNA on the U spot. So eventually we're going to have the start codon, which is a codon here. After the process of DNA transcription happens, the DNA transcription goes along the binding site, the proteins break off through enzymes or substrates, and it gets cleaved off into little segments that your body can read for genetics appropriation. So we have the OG, which is the start, then we're going to have CGG, which is going to be an amino acid, that nucleotide, the monomer, which eventually makes the nucleic acids. You're going to have the CGG, which is arginine, and then you're going to have leucine, and the sequence can go all the way on until you have another stop codon. Once that stop codon occurs, that's when it cleaves back off, and then the binding and the substrates, the phosphorus groups, don't occur as often. Alright, so this completes my project on the hierarchical structures of macromolecules. I hope you enjoyed it. I know it might have kind of been bland, but I spiced things up in life. Thank you for watching, and have a great night.